So it's my pleasure today to introduce our latest work, which is basically looking at novel ways of identifying traits that co-occur with chronic obstructive pulmonary disease. And this is work that's been led largely by Eugenia Golovina. So chronic obstructive pulmonary disease, or COPD, is a heterogeneous condition. It basically affects the lungs, but it's really accompanied by a lot of other conditions, and they include diabetes, lung cancer, anxiety, asthma, all these sorts of things that you see on the slide. And they affect different body organ systems, and it's really important to understand that. So genetic studies have identified the common single nucleotide polymorphisms that are really linked with COPD. And also with the individual co-occurring conditions. And so this is pretty cool, it works really nicely. The problem is that we don't know how that risk is associated with these predominantly non-coding SNPs, and we don't know if or how shared genetic risks for COPD and the co-occurring conditions interact. And that was really the driver for our study, was to try and understand how these things might be interacting in order to either define different subtypes of COPD or to show how it basically develops. So to address this, we really just set about integrating information that was available on common COPD associated genetic variants, lung-specific 3D genome structure, gene expression, and protein-protein interaction networks. And really what we tried to do was to build a lung-specific gene regulatory and protein-protein interaction network. And so we did this two ways. The first way was that we were using a pipeline that we've used quite a bit before, which is one that we developed called Codes3D. And what that basically does is it allows us to take the COPD associated SNPs, and there's 263 of those, effectively to work out using three-dimensional structure, basically what genes they are associated with or they have an EQTL with. And that works very simply in that we can see what the region the SNP is marking interacts with. We can identify a gene that could be in cis or trans within a chromosome or trans between a chromosome, and we then can basically check between that SNP and the gene that it interacts with whether or not we have an EQTL. And if we do, then that there is a putative function for that SNP. But we also took the 47 million common SNPs that are present in GTEx, and we ran those through the same pipeline to create a whole lung-specific gene regulatory network. And this was using lung-specific EQTLs and also lung-specific Hi-C data. So what we were able to do then was basically to combine those two things. We were able to take the COPD associated E genes that we identified and work out what other regulatory elements were interacting with those genes within the lung gene regulatory network. But we were also able to expand out, and by using the proteins that they interact with and effectively creating a protein interaction network, we were able to take the proteins that were encoded by those COPD associated E genes, determine which proteins interacted with them in the first step, and then which ones interacted with those in the second, and effectively to create levels of protein associations. By doing that, we were able to then work out which EQTLs were regulating those genes within the lung network. And doing that meant that we were able to get all of the EQTLs, all of them, within reason, the EQTLs that were controlling the different genes on the different levels, or that encoded the proteins on the different levels of these networks. Then we were able to take those EQTLs and run them through the genome-wide association catalogue. And by doing that, we were able to identify the comorbid traits that were basically interacting with COPD, either directly or indirectly through other proteins. And this pipeline that we've done here, we made up here, is called multimorbid 3D. So doing this basically enabled us to do two things. One was to identify the genes that are associated with or whose transcript levels are associated with the COPD associated SNPs, and we did that. We found 107 COPD associated genes, and these genes were, encoded proteins that were enriched in biological pathways for the regulation of actin cytoskeletons, insulin signaling, resistance, focal adhesions, all sorts of things. There were 9 COPD-associated lung-specific protein-protein interaction sub-networks that we identified, and they included things like the nicotine receptors that we see here, cron A3, cron A5, and various other really interesting small protein interactions in the lung. Taking this information and actually looking at what was occurring or what was interacting with these COPD-associated EQTL genes, basically by doing that we were able to see that there were a number of different conditions that were associated. Now here we have the network map, so we have level 0, which are basically the pleiotropic genes that are affected by COPD-associated SNPs. And then we have the proteins that interact with these directly on level 1, the proteins interact with those on level 2, the ones that interact with the level 2 ones on level 3, and level 4 interact with level 3 on level 4. And basically what you see here are the EQTLs, the numbers of EQTLs, and the p-values, adjusted p-values basically, for the trait that we're seeing here. So we see different traits that are interacting. So on the pleiotropic level, level 0, we see things like Parkinson's disease, neuroticism, lung functions, as you would expect, COPD, a very range of those as you see here, brain volumes, asthma, etc. And then on other levels we see different things. So we see atopic asthma coming on level 1, we see Alzheimer's on level 1, and we see various other conditions which are basically linked on levels 1 and 2. Now, we looked at some specific subnetworks, and so we looked at the NUPR1, MSL1, SGF29, and MOCS2 protein-protein interaction network. And that basically involves a number of genes, or 4 genes, and those 4 genes basically are associated with EQTLs linked to COPD and lung functions. Now, the 4 genes interact with 3 other genes on level 1 that are associated with post-bronchodilator if forced expiratory volume, atopic asthma, and Alzheimer's disease. And this is really cool. And those 3 genes that are on level 1 are basically CAT8, RUVBL1, and SUOX. So this is a really nice example of how this expands things out and shows you some of the interactions that may be occurring and contributing to COPD and other conditions that occur with it. Now we also did a similar thing for the SSH2 test2 subnetwork. And so this is a protein-protein interaction subnetwork that was associated with EQTLs linked to COPD, lung functions, brain region volumes, and coronary artery disease. And this occurred on level 0, so these are pleiotropic genes, SSH2 and TESC2. And you see here the EQTLs that are associated with them. Now SSH2 is regulated by a region on chromosome 17, and TESC2 a region on chromosome 1. Now, notably, these two, there were no trait EQTL gene associations identified within level 1 for this particular subnetwork. So that's a little bit different from the one that I showed you just a minute ago. So basically what we were able to do was to identify target genes that are associated with COPD-associated EQTLs that may interact to connect COPD to co-occurring conditions. We were also able to show that the results provide multiple new avenues for future investigation of underlying biology and diverse clinical presentations of COPD. And the way they do that is that they basically show both the genetic variation, the biological interactions in terms of protein-protein interactions that are occurring in this example, the lung, and may actually help to stratify different subforms of COPD. Now, we think that confirmation of these connections will actually suggest potential therapeutic COPD markers for follow-up and patient stratification. So I'd just like to acknowledge that this work was funded by the Dynes Family Charitable Trust, and then there was also a lot of funding that was provided for the New Zealand eScience Infrastructure and Genotype Tissue Expression Project, which was supported by the Common Fund. If there are any questions, please either tweet or go through and email myself or Eugenia, and thank you very much. I hope you enjoy it.